Our next speaker is Dr. Andy Badrock, a molecular geneticist who specializes in using zebrafish to better understand human disease. His current research focuses on LCC and how laboratory models can help us uncover what is happening at a cellular level and identify possible paths towards treatment. Dr. Badrock is joining us virtually today, so please join me in welcoming him.
Okay, thank you, Ashley, for that kind introduction. By way of introduction, yes, I have been working on LCC from a molecular perspective, and I've been working in the Crow Laboratory for close to 9 years now and trying to understand LCC genetics. And I do want to apologize in advance. This is a very science-heavy presentation, but I'll endeavor to make it as accessible as I can. So just very briefly to touch on some of what's already been said. So U8 is a non-coding RNA. What it does is it removes some spacer sequences called the 3' externally transcribed spacer sequence and the internally transcribed spacer sequence 2 from a long ribosomal RNA. And what this does is essentially generate a ribosomal RNA called 28S as well as 5.8S. These then combine with ribosomal proteins to form part of the ribosomal subunit. And then, as has been mentioned, ribosomes then translate mRNA into protein, and they're very important really for every cell in the body. As has also been mentioned, what we know is that it's largely biallelic mutations that cause LCC. And what we mean by that is variations that essentially are different from each other. And so this then typically results in calcification and cyst formation. So one of the problems we have as scientists is we struggle to understand how sequence variations in a non-coding RNA actually affect its function. And so what I did was to use the zebrafish to try and address this challenge. Now, in a human or a mouse, you only have one SNORD118 gene. The zebrafish are somewhat unusual in that they have 5 copies of U8. However, it's only this U8-3 that's expressed very early in development. And so I used a technology called CRISPR-Cas9 to delete that gene from the genome. And what we create is this zebrafish mutant that has no functional U8-3 at all. And it's characterized, and what it essentially develops compared to the wild type embryo after 2 days of growth is swelling in the brain. So this is labeled by the white arrowhead. These are melanocytes, pigment cells that migrate over the yolk of a zebrafish. These no longer migrate, and that's labeled by the orange arrowhead. And then this structure here called the yolk extension, which forms part of the yolk which the zebrafish feeds off for the first 5 days of its growth, fails to form, and that's labeled by the red arrowhead. And then what I was able to do was to inject a precursor form of U8 and of human U8, and essentially it's a human U8 with the short 3' extension that Jamie mentioned earlier. When I inject this human U8, I'm actually able to rescue this mutant. So what we see here is there's no longer any swelling in the brain. The melanocytes move over the yolk, and then we have this yolk extension structure is completely rescued. And to prove that I've successfully injected this human U8, I also co-inject it with a mRNA essentially taken from a fluorescent coral called MK2, and this turns the whole zebrafish fluorescent red. So as I mentioned on the previous slide, what U8 does is it makes 28S, or a thing called 28S. And we can see in this little zebrafish embryo that has no U8-3 function, there's much less 28S compared to its wild type sibling. But then when we inject the human U8, we can rescue this. So we don't rescue it completely. So we have no function for U8-3 here. We have some function for U8-3 here. But it's quite remarkable that the human gene can also function in a fish. And another, in this case, mRNA or an RNA that we look at is called interferon phi1. This is a gene essentially that gets highly upregulated when you have a very severe translational stress, when none of the ribosomes are working, which is what's happening in this embryo shown here. And then the human U8 completely rescues that. So there's no difference between the wild type or this rescued mutant. So this is just an independent way for me to show that, yes, I am actually rescuing the U8-3 zebrafish mutant and the human U8 is functioning. So, of course, the next question I wanted to ask was what happens now when we look at the sequence variations that cause LCC? Will we see this same degree of rescue? And I'm now going to present what is a very complicated figure from the paper that we've just heard back from. So we've submitted that to a journal and we now have reviewers' comments that we're trying to address, and hopefully this will come out formally very soon. But what we see is that for 50 different variations that have been linked to LCC, the variations that are labeled in black are the ones that they actually rescue the zebrafish mutant, and the ones in red do not. And what we see is this is what we call a heat map of expression for 28S. So the more blue it is, the better the increase in 28S. And we see that always occurs when you're rescuing the phenotype, the defects that develop in the U8-3 mutant, and then you suppress this interferon. And then we see the opposite with the LCC causative variations that don't rescue the mutant. But the take-home message is, top panel here is showing sequence variation in the human population. And we tend to find that where variation is low, the LCC causative variations don't tend to rescue the mutant, but where sequence variation is much higher, they do. And we think this might be useful for clinicians and potentially developing algorithms that can predict whether or not an LCC causative mutation is mild or more severe. But the take-home message really was that the vast majority of patients seem to have one functional or one non-functional variation. And this fits with what we call the biallelic nature of the disease. So one of the really surprising observations from this rescue with the human U8s was, even though I'm injecting the same amount of U8 into a wild-type embryo as the U8-3 mutant, I see much higher levels of expression in the mutant compared to the wild type. And we were wondering whether or not this is an attempt at, because we have a partial level of function, we were wondering if this was an attempt at compensation. And if we could understand this, perhaps we could stimulate increased function in patients with LCC. So I was concerned it was a bit of what we might call an artifact of me injecting this U8 into a zebrafish. So I also made a partially functional mutation in the Zebrafish U8-3. I won't go into the nature of the mutation because it's quite complicated, but the take-home message is that we see the same thing, that in this partially functional mutation with much higher levels of expression than when that mutation is expressed in a wild-type animal where ribosome biogenesis is normal. And we can see this with a different technique. This is RT-PCR, where we can actually see this is the partially functional mutation. This is wild-type allele. And we can see only in this mutant are we seeing that much higher levels of expression. So because this potential compensatory response is occurring in transcripts that I inject, that's known as a post-transcriptional mechanism. And one way you can have such a mechanism is if you modify U8 with, I guess, chemical modifications, and if you can change them to change their interaction with proteins and their function. So I wanted to understand whether this was occurring. And so this is a diagram of the structure of U8, and I've color-coded a bunch of different structures that have different functions. I won't, again, go into it in too much detail, just to say there's a database that actually tells us that U8 has 5 modifications at position 21, and 108 is a modification called pseudouridylation. And then at position 53, 105, and 112, there's a modification called M6A or N6-methyladenosine. So I really want to understand possibly these modifications changing the U8-3 mutant compared to its wild-type sibling. And one of the questions we actually had from reviewers only last week was, you don't know that these modifications are happening in a zebrafish. And so we used a technique called nanopore. And what that does is it just allows you to sequence directly a RNA, and it has the potential using electrical currents to detect these pseudouridylation and M6A modifications. The first thing we saw was that this modification at 108 is very highly detected. It's apparently 90% of U8 injected into a zebrafish is modified there. And it doesn't really change in the U8-3 mutant when you inject the human U8. So we don't think this is necessarily the cause of the increased expression I've just been talking about. I'm going to skip over that point. It's a bit complex. But what I wanted to do then was ask myself, given my zebrafish model, can I encode a mutation here? Can I block this pseudouridylation? And then what will happen? What will happen to the ability of U8 to rescue the U8-3 mutant? And so that's shown in this experiment here. Just to repeat, this is a U8-3 mutant. It has no function for U8-3. It has this fluid in the brain. The melanocytes don't migrate or move over the yolk, and it has no yolk extension. Human U8 rescues all of these features. And what was really interesting is that human U8, where it can't pseudouridylate, that nucleotide rescues even better. So we have the melanocytes move much further over the yolk, and we can even measure the eye size of these zebrafish embryos and show that a mutant with no function has an eye that's 50% smaller than its wild-type sibling. When you rescue with human U8, it becomes around 30% smaller, but it's only 15% smaller when you rescue with this human U8 where you can't pseudouridylate position. And so we're thinking that potentially if you can block this modification, that might be a way to enhance U8 function. That might be something that could be done with the right drugs in a LCC patient. So I also want to look at this M6A modification. Of the 3 modifications, essentially we detect 2 of them, one at 53 and one at 105. But again, it doesn't change in the U8-3 mutant when you're expressing the human U8. And ultimately, we think whatever is increasing the expression of U8 in the U8-3 mutant, it's not down to changes in modification, but we still wanted to understand if we block these modifications in the zebrafish, what would happen. Again, I'll skip over that. And essentially, not much happens to the ability of U8 to function if you block different M6A modified nucleotides. We don't have any hindbrain swelling in the mutant. We can rescue the melanocyte migration, and we have a yolk extension, whereas you wouldn't in an unrescued mutant. But what we do see, because when we look at the expression levels of these U8 molecules that we've injected, the wild-type U8, as I was saying earlier, has this higher level of expression in the mutant compared to its wild-type sibling, and this increase in expression is reduced. So there is some effect, at least on stability or expression levels, which is interesting. So key question for us as well in the lab revolves around trying to understand why some LCC patients have milder forms of the disease than others. And one of the thoughts that we had was maybe that some of the people with milder disease might have a sequence variation that blocks some of these modifications. And so I went through our cohort, I looked at all the sequence variation, and what was really surprising, and this is a very complicated table, but it appears that pretty much the entire cohort can't make the M6A modification at position 105. So we were only able to interpret this because of the zebrafish data. So there's only 3 patients that can't do this or can modify 105. Most of them have 2 copies of the 105 A to T, but because we knew which sequence variation that causes LCC, which one functions and which one doesn't, we could see that on the functional allele, on the functional U8, it couldn't make a modification. So we are wondering whether or not, in addition to looking for what the clinic might refer to mutations, do we also need to look at the sequence variation at position 105? And so to conclude, the functional states from the zebrafish seem to support what we would call the clinical genetics, and it suggests that the vast majority of LCC patients, they have one functional mutation and one non-functional mutation. And from a therapeutic perspective, if you were trying to increase U8 function, you would focus on functional one. We also, for the very first time, look at the effect of mutating modified nucleotides of U8, and we see that this affects not only its expression, but also its activity. And we are wondering if this N or nucleotide 105 A to T variation is important for the development of LCC. And when we're being contacted by clinicians, when they're asking us about whether or not they've detected mutations, we're also asking them to look at this sequence variation. And so far, it's the same pattern is happening in new patients in the clinic, as I've just shown on the previous slide. And of course, if we want to make disease models of LCC, then taking this into account may actually be quite important. So, future directions. One of the things I'd like to explore is whether or not blocking pseudouracilation at position 108 could be used to enhance U8 function in LCC patients. And because this position 105 is quite close to the 108, I'm wondering whether or not these two modifications might communicate to each other, and that may explain to us why we see this variation in patients with LCC. And this is something I'm assessing with nanopore sequencing, hopefully in the next week or two. So with that, I'll just finish with acknowledgements. I'd very much, in terms of funding, like to thank Mandy Vine and her family, the Great Ormond Street Hospital, LifeArc have funded this project at various stages, as well as the MRC. I'd like to thank my supervisor, or the boss of the lab, if you like, Yannick Crow, also the LCC patients and their families, and Professor Emma Thompson, who has helped me with the nanopore experiments, and that's been very informative. And so with that, I'm happy to take questions.
Hi, Andy. That was a great talk. Thank you so much.
Yeah.
One question with these modifications, have you seen whether any folks where there is discordance in the genotype-phenotype association have differences in the two modifier sites?
I'm not sure I understand
So,
the
for
question
example, if
correctly.
there are siblings who are discordant by phenotype,
Yeah.
would you anticipate that modification of the sites might be part of why they're different?
So not at this stage. So that's what I was really hoping for, that maybe that might be the case. It pretty much all we saw was the entire cohort, regardless of severity of disease or genotype, seemed to lack the ability to make this modification. So we think it's something that I guess clinicians should be aware of. And it would be, I guess, as more patients come to light, perhaps we will see genotypes where that modification isn't blocked, and that might tell us something. What I don't know at the moment is whether this sequence variation is protective or whether it's making people susceptible, and then whether or not that's something you can manipulate with some form of RNA therapy.
Hi, this is Jen Orthan. Thank you for this really interesting talk and model that you can really capture this early developmental phenotype with how you set this up. And I'm wondering, do you think all cells are equally dependent on these mechanisms? With the zebrafish, you're capturing these very early findings, but is this more central nervous system, or how do you narrow it down which cells are more dependent on this ribosomal formation with U8?
So I think one thing about my zebrafish model is I'm working with much more severe mutations than I think cause LCC. So in terms of the defects I'm seeing, it's useful for me to assess how U8 functions, but I wouldn't say I'm modeling LCC. I'm not seeing calcification. I'm not seeing cyst formation in the brain, these sorts of things. But I guess in terms of patients, so given that modification of U8 might be important in development of LCC, one thing we know is that RNAs tend to be more modified in the brain than any other tissue. And that could potentially explain why neurological symptoms occur when you have these sequence variations in U8. But equally, it could just be what I would call an artifact of biology. So most tissues in the body they might make more U8 they need. So that when you have sequence variations in them, that doesn't affect those tissues, but there might be a subset of cells in the brain where that's not the case. So from my point of view, I would say maybe the U8 promoter isn't very active in cerebral vascular tissue. And therefore, when you have sequence variations, it's that particular tissue that's affected. But at the moment, that's all very much speculation.